Okay, today we're gonna talk about ChIP-seq. So let's first do a recap. We still have a little unfinished business from the last lecture. We know that genes are regulated because transcription factors recognize specific sequence patterns to bind to the DNA, then it can regulate nearby gene expression. And in order to find those transcription factors and where they bind and figure out, what's the effect they have on the nearby gene, we, in the last lecture, talk about the different computational approaches for motif finding. These methods are okay when you look at the yeast or bacteria, and maybe even C. elegans. But by the time you look at human mouse, things are usually quite complicated. We mentioned that regulatory sequence that are bound by transcription factors, it can be quite far away from the target gene, and the sequence can be short or long. The motif can be short or long, and you can already start to see them appear in combinations with other transcription factors or clusters of the same transcription factor repeating itself, just so that the transcription factor will sure to bind that location and regulate the nearby genes more tightly. In most of the case, we don't have enough of these gene expression perturbations to give you a very good set of genes that are co-expressed. By the way, the genes that are co-expressed in a cluster, we hypothesize that they may be co-regulated, but they are not always the case. Sometimes when the signal is here, this transcription factor regulates something, but one of its child also is a transcription factor and they regulate other things. So you would see the uncles and together with the children together, but you hypothesize that they share some similar signals. And so in order to do motif finding in higher eukaryotes, people look at, first of all, over the last 20 years, there are more and more known transcription factor motifs that are experimentally derived. People start to look at comparative genomics to look at sequences that are conserved. They start to look at the motif modules for motifs that are clustered within a short range of each other. And then they do ChIP-seq or ChIP-seq. So let's go over each of them. PBM, we went through last round, so there are experimental approaches to get the motif information for some transcription factors. And one thing that people notice is that usually the same family of transcription factor, they GATA1, GATA2, GATA3, GATA4, GATA5, they very often recognize quite similar transcription factor motifs. They might be expressed in very different cells. They might also interact with different partners and regulate different target genes, but the specific sequence pattern they themselves recognize can be identical or quite similar. With comparative genomics, this is something that people do. So UC Santa Cruz is the genome resource. When the first human genome was sequenced, there needs to be a genome browser for people to download the different gene annotations, to visualize the sequence, to look at how genes reside in the genome. And so that's resided in UC Santa Cruz. And they have developed this tool called phase conservation score. Basically, they look at whether the sequences are under strong evolutionary constraints. Initially, there was like human and mouse, and they're like, now you can see here, this is different vertebrates, mouse, rat, rabbit, dogs, armadillo. In fact, because genome sequencing now is so inexpensive, people are sequencing hundreds or thousands of different species, and you can align the sequences together. Especially, usually you align them by the genes, because genes are still much more similar between the species. And once you can align the genes, you can look at the sequence near in front of the gene, in the introns of those genes, and also after the end of this gene. And you can do this type of multiple sequence alignment. And this is an example of some alignment. You can see these sequences, they appear. So by the way, we're looking at the main track is human, and then you are comparing with mouse, rat, rabbit, and dog, and so on. So if there is a black mark here, it indicates that sequence at least is also present in mouse, and also how similar that is to the human sequence. Whereas in chickens and tetrodon, those sequences don't really appear. And so you can see there is a conservation score in here. If it's very high, it means, you can see here, this is very deeply conserved, which means this sequence is present and also quite similar in all the other species. Whereas you can see this region is only present in human but doesn't appear in the other species. So it's a not very conserved sequence. So phase conservation score is between 0 and 1. 0 means this is not conserved at all. 1 means it is very, very conserved. But there are different tracks of this phase conservation score, because people sometimes are interested in only conservation within all the primate species, orangutan, gorilla, you know, those. And then there are people who are interested in all the conservation within, say, mammals, right? And then you can go all the way to fish, right? So you can download different tracks. They tell you how many species are used, or which are the species used to generate the alignment and calculate that conservation. And so this information can be very useful. You can imagine that if we cut off at, say, 50% conservation, so this, remember, is a score between 0 and 1. If we cut by 50%, 0.5 on the conservation score, you are not going to eliminate just 50% of the sequence. You can see if you cut off at this point, you're going to eliminate probably 90% of the sequences that are not conserved. That is not to say that the things which are not conserved are completely useless. For example, if you want to look at some genes related to ovarian cancer, and you want this thing to be completely conserved in fish, it's going to be harder because the fish does not have ovary. Well, no, actually they do have egg, but it's, or breast, or, you know, like, you know, depending on the level of regulation, you may or may not need the conservation to be that deep. Sorry, fish does have ovary. They don't have a mammary gland, right? So that's the difference. And so you can use conservation to eliminate sequence so that if you have to look far away from the transcription start site, you don't need to look at all the sequences. You can narrow down your search space. So there are some very interesting studies generated on this. For example, this is a region in the genome that's a gene desert, which means the nearby genes are really, really far away. And suddenly there's a region, there are actually islands of these little regions that are a few hundred base pair long. And if you look at their conservation, from mouse, or from human to mouse to chicken, frog, fish, it's really ultra conserved in all of the vertebrate species. So I want to ask you some question. Is conserved sequence functional? If you have something like this that's really conserved, if it is this conserved, it's got to be functional. Just because we don't know what they do doesn't mean that they are not functional. Yeah, so there's a reason for this thing to be ultra conserved, right? And so, yeah, so some of them, of these conserved regions, are in the exons enriched in RNA sequence processing. Those are in the coding regions of the RNA. And sometimes it's important for splicing. But the really interesting thing are sometimes even in gene desert, there's in the middle of nowhere, right? That suddenly you see this region that's really conserved in the species. And then there are some experimental work that are conducted to test this. So we mentioned that conservation does not mean function. If something is not conserved, it can still have function. But if something is super conserved, it's got to have function. And this is some example that people have done. Interestingly, the sequences that are super conserved throughout the species, they are usually related to development, because, you know, when we are embryos, whether it's a pig or a rat or a human, they all kind of look like little fish, right? So those early development genes regulation has to be tightly controlled, and those are very, very conserved, and you do see that happen. And so, for example, in order to see what this region does, you know, you could insert this into or label the genes near this region and see. So you put a gene, like a reporter gene, that give out a blue marker, and you ask when is that gene getting activated and potentially regulated by the sequence. So you can put a blue marker gene near that region and see whether you have this sequence or you delete this 200 base pair sequence, that marker gene will be different. And so in the embryos, you might be able to see, oh, this appears in the brain or in some heart of the early embryos. That will give you some idea. Roughly what tissue is this region important, and you know what, and to really show that it has a functional role. So you can mutate this in different ways and see whether that changes the phenotype. Okay. Another interesting study that people have looked at is human accelerated region. So there are regions in the genome, for example, in this example, this is the human sequence. And if you look at the chimpanzee, rhesus monkey, mouse, rat, dog, chicken, they are quite conserved. And suddenly in human, all these bases are changed. There seems to be a positive selection, meaning that this particular nucleotide, once it changes, somehow giving human a much better evolutionary advantage. And therefore, they are being fixed into the genome. And you wonder what they are. And so scientists have also done this type of research. And for example, they found that this particular sequence is actually very important for neurodevelopment because of these basis changes, humans are much, much smaller. In the same study, they have also shown that the accelerated regions, the sum sequence are very different in human, because I guess human use the sum much more differently and much more flexibly, rather than just climbing trees we type. So these are the interesting things. So I would say not conserved doesn't mean not functional, but conserved are definitely functional. And you can use conservation to eliminate some of the random garbage, and so you can enrich for your signals to look for motifs. And then we mentioned, we already see this in yeast. Very often motifs are clustered in a region. Usually the cluster is 150 to 200 base pair long. And suddenly within, you can see what in this particular study, they were looking at a 100 to 2000 base pair long region. But very often those modules, we call them a transcription factor module, is usually one or multiple transcription factors bind in a concentrated way within a couple hundred base pair sequence. And that module is very important. You can use sequence conservation first to eliminate a lot of sequences first, and then within each of those fairly, a little bit more conserved sequence, you can ask, do you see the same motif appearing over and over again? Do you see a combination of different motifs and things like this? And so there is a study generated on this, and they can eliminate a lot of the irrelevant sequence in the genome. And interesting, a lot of the regions they identified later was proven to have very important functional roles. And so this kind of are some landmarks. Suddenly you see a lot of things happening there. That's an important regulation region. And if you do that for motif finding, to look for motif clusters, you are more likely to get some interesting result. But despite all of this, I would say transcription factor motif finding is just not very easy. In fact, when I came here in 2002, as I mentioned, our collaborators just have some mouse experiment when they treat with the mouse with 2 drugs, and then they see 2,000 genes upregulated. And to do motif finding with that, because at that time, the upregulated genes could be many different situations. We don't have a good cluster of genes. It's very, very hard. The savior of my career is this technology, which is called ChIP-seq. Now it's called ChIP-seq. The first ChIP stands for Chromatin Immunoprecipitation. The second part is now high throughput sequencing. So basically, it's a way for scientists to very quickly look at how a particular transcription factor binds into the genome. At the genome-wide, all the locations you can investigate, and you want to ask, where does this one transcription factor binds throughout the genome? The experiment worked like this. So each of the rectangle here is a sequence on the DNA. Usually we call those cis element. It means the DNA on the genome which is being recognized by the factor. And each of the other shape is a different transcription factor. The red star is one transcription factor. The green circle is a different transcription factor. You can see they are called the trans factor or transcription factors, right? Trans factor just means usually these are proteins and they bind to the cis element in a sequence specific way. And even in naturally occurring, you just grow a cell in a petri dish or you take cell from some individual tissues, there will be naturally occurring transcription factors that bind to the cis element. And so in the chromatin immunoprecipitation, the very first step you do is to do cross-linking. We use formaldehyde to cross-link the protein-DNA interaction. There's going to be a bond formed between the transcription factor and the DNA covalent bond. So it's very stable. They are now stuck together. And then there are machines that can sonicate, use sonic wave to break the DNA into shorter pieces. Because we have done the cross-linking, the transcription factors are still attached to the DNA that they bind to. And then if we are interested in a particular transcription factor, and if that transcription factor, you have an antibody. In this case, we are interested in this red star protein, and there is already an antibody that has been raised, which uniquely recognizes the red star protein. You can add the antibody to this solution. You basically, you break up the cell, the DNA, the chromatin is already sheared into small pieces, and you use the antibody to interact with all the red star protein. And this antibody can have a little label at the end that you can spin down with either magnetic bead or something, right? So anything that does not interact with antibody can be washed out. Anything that is, say the antibody is attached to a bead, then you can spin down the bead and they enrich for all the red star protein, as well as the DNA that's enriched, that's bound by it. You do notice that sometimes, so antibody recognition is not 100%. It's probably, well, you can think it is 100%. It means that it will probably still make a mistake 1 in 1,000. That's already a very, very specific antibody, but because in the genome, in the cell, there are so many other proteins around, occasionally it will recognize some wrong guys in here. And also, sometimes there are some random piece of DNA that just stuck to the bead or stuck to the tube of your experiment. They also get pulled down. Hopefully those sequences are not consistent. By the way, we do this experiment in usually hundreds of thousands of cells. In the early days, we have to do this experiment with usually a million cells. So it does require quite a bit of starting material. And so hopefully those non-specific piece of DNA and also those non-specific binding will not be repeated in the different cells, you know, whereas this antibody binding to the red star protein and binding to this piece of DNA is consistent in most of the cells. We are hoping that these random noise will, you know, won't be consistent. And after we have enriched for this, there's a reverse cross-linking. You usually just heat up you boil this solution, and then the protein will break off from the DNA. And then you isolate the DNA, and at this point, you can amplify it a little bit, then you can do high throughput sequencing. Okay, so with RNA-seq, you can do RNA-seq with very few cells because each piece of DNA is able to produce a lot of different copies of the RNA. Whereas for ChIP-seq, because you are using a protein to bind to DNA and every cell just only have 2 chromosomes. And so you do need a significant amount of starting material to do this experiment. And so at this point, there would be PCR amplification and then sequencing adapter, then you put it in high throughput sequencing. This is coming from a different review paper. They also show, you know, proteins binding to the DNA, you sonicate into small pieces, and you use the antibody to recognize the protein of interest. You enrich for those DNA bound to that protein of interest, and then you try to do high throughput sequencing. So this is what the signal looks like. Because in different cells, as you use the sonic wave to break up the DNA, supposedly this is one region where there is a transcription factor motif and the transcription factor is binding to this location. In many different cells, the transcription factors are binding to the same, say, chromosome 1 location 10,000. But then when you try to sonicate the DNA into small pieces, the breakage location is kind of random. So sometimes it breaks like here, another time it breaks like that, another time you break that like this. And at the end, when you sequence, by the way, early day when people do ChIP-seq, sequencing is quite expensive at that time. By the way, high throughput sequencing for Illumina, the very first paper published was not on RNA-seq, it was on ChIP-seq, because it was just the count that's important. And at that time, it was, I think, only sequence of 30 or maybe even 25 nucleotide for each fragment. And they can only sequence one end because sequence the other end is twice as expensive. And they just, okay, randomly pick one end to sequence. And the one thing they notice is that you probably see here, if you sequence from this end, remember DNA is double-stranded. If you sequence from this side of the binding side, then it will map to the Watson strand versus the Crick strand, right? So if you sequence from this side of the binding, the sequence will come from the Watson strand. Whereas if you sequence from the right side of the transcription factor binding, you will sequence from the Crick side, right? So you will see basically when you look at many different cells, we're sequencing all of the cells together, right? What you will see is that in some regions, suddenly you will have kind of like a peak or even two peaks. Half of those are coming from the left and they all map to the plus strand on the DNA. Whereas the right side, it's all mapped to the minus strand on the DNA. And very often that means the transcription factor is binding right in the middle. Okay. By the way, there is also a control experiment in ChIP, and it's usually using input control. You still do the cross-linking, you still shear them into small pieces, but then there is no antibody enrichment. You just directly decrosslink and get this whole short fragment, sequence them out into the genome. And you will get the idea if there are some biases, or some regions are easier to sequence than others, you can see that directly. And so that's a control experiment. Okay, and so very often ChIP-seq is done with 1 to 2 chip and 1 to 2 input controls. You can publish even a single ChIP-seq without control if the signal is pretty good. It's not, I would say probably half of the published ChIP-seq data don't even have replicates, which is still okay. All right. Yes, question.
So how do you decide if it's does the transcription bind, how much bases the transcription factor need to bind to? Does that dictate how long?
I see. So the question is, how long are these transcription factors bind? And does that indicate how broad this thing is, right? Different transcription factors bind differently. In mammalian genomes, we usually see transcription factor binding from even 5 to 6 nucleotides to as long as 16, 18 base pair long. Most of them are probably 15, 16 is kind of the rare, is not the norm. The norm is, yeah, I would say 6 to 10. So actually for motif finding, very often we try something like you try a 6, you try a 10, you try a 14, and you just see whether it gives you something. Okay, and so the peak, what kind of shape does it have? It depends a little bit on the transcription factor binding width, but also depends on how harshly you sonicate. If you sonicate for longer time, the fragment will be shorter. But if you don't sonicate as much, the fragments will be longer, then this peak will be a bit broader, right? So in the ChIP days, people don't sonicate as much. They still have fragments that are over 500 base pair because they need many probes to cover that region in order to call it. But nowadays with ChIP-seq, people usually sequence down to about 200 base pair long. When it's too long, when it's too short, sometimes you can't sonicate anymore. Usually, I think even if you were to sonicate like crazy, you probably still see a band around 100. Okay. So that's the ChIP-seq experiment.